most of my research works in the idea of building computational models and software for analyzing microbiome data and then applying that in exciting ways to ask important questions. And so what this slide is really showing is our approach to microbiome work. And it really is a focus on both the host and the microbiome. On the one side, you're looking at microbiome profiling. We've developed a lot of tools for taking sequencing data, for example, RNA or DNA sequencing, and profiling the microbial content. And then on the right side, looking at the host, for example, host gene expression, and what pathways are active in those genes and pathways are active in those hosts, and then linking, for example, pathogen abundance with host pathway expression, and maybe not even pathogens, but the microbiome with host mechanisms, and how can we then connect those? Next slide, please. Exciting to say that we've developed this tool that we call Animalcules. It's an interactive microbiome analysis toolkit. It helps you analyze your data. Here's some screenshot examples. This is available, and our paper just got accepted. It's going to show up in Microbiome, the Microbiome Journal. And hopefully this makes it easy to analyze your data, put it that way, in a point-and-click manner. Okay, next slide, please. Jumping to, and I don't have much time to talk about this, but one exciting area, we're applying this in a lot of areas, but one exciting area that we're looking at is in lung cancer, and looking at and seeing if we can use microbes to predict whether or not do diagnostics for lung cancer, or even look at the progression of lung cancer. Next slide, please. Next slide again. Click it one more time. And then we're working with Daniel Segre as well, looking at microbe interactions in this case. So what microbes are interacting with each other that influence lung cancer. Next slide, please. And then also looking at microbe host interactions. And again, for the sake of time, don't have a lot of time to go into this one. Next slide, please. But what we're looking at are, you see this heat map, is you see human pathways by microbes, and these are microbes that potentially activate human pathways, and then connecting that to basically the development of cancer. For example, PI3 kinase has been established as an early event in lung cancer, and it's also been associated with the presence of microbes such as Streptococcus in the lung. And then it's been shown to be reversible by chemoprevention using agents such as minoxidil, which turns out to be a microbial metabolite. And so we're actually digging in and actually looking at these microbe by drug by host interactions and trying to use that to essentially drive our analysis. Next click, please. And from this analysis, we've looked at these other mechanisms here. For example, Corynea bacteria is also associated with PI3 kinase or glycolysis or reactive oxygen species, for example. And so these are all potential cancer-driving mechanisms that are associated with microbes. And so we're in there studying that.